Good day. My name is Joshua Glenn Piesta, student of Bachelor of Secondary Education, major in General Science at Philippine College of Science and Technology. Our lesson is mitosis and meiosis. So let's start with the question. What is mitosis? Mitosis is a process of nuclear division in eukaryotic cells that occurs when a parent cell divides to produce two identical daughter cells. During cell division, mitosis refers specifically to the separation of two duplicated genetic material carried in the nucleus. Mitosis is conventionally divided into 5 stages, known as prophase, prometaphase, metaphase, anaphase, and telophase. So let's start with the first stage. Prophase is the first stage of cell division in both mitosis and meiosis, beginning after interphase. DNA has already been replicated when the cell enters prophase. The main occurrence in prophase are condensation of the chromatin, reticulum, and the disappearance of the nucleus. The second stage is prometaphase is the second phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During prometaphase, the physical barrier that encloses the nucleus, called the nuclear envelope, breaks down. The third stage is metaphase is the third phase of mitosis, the process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. There is an important checkpoint in the middle of mitosis called the metaphase checkpoint, during which the cell ensures that it is ready to divide. The fourth stage is anaphase is the fourth phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. The separated chromosomes are then pulled by the spindle to opposite poles of the cell. Then telophase, the fifth one. Telophase is the fifth and final phase of the mitosis, the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. Telophase begins once the replicated pair chromosomes have been separated and pulled to opposite sides or poles of the cell. For meiosis as well, we will also start with a question. What is meiosis? Meiosis is a special type of cell division of germ cells in sexually reproducing organisms used to produce the gametes such as sperm and egg cells. It involves two rounds of division that ultimately results in 4 cells with only one copy of each chromosome. There are 6 stages within each of the divisions, namely prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. In this article, we will look at the stages of meiosis and consider its significance in disease. So the first was prophase 1. Prior to prophase, chromosomes replicate to form sister chromatids. Prometaphase 1, the second in meiosis. Spindle fibers attached to the chromosomes at the points along the chromosomes called centromeres. While this is happening, the chromosomes continue to condense. The third in meiosis was metaphase 1, maternal and paternal version of the same chromosome, homologous chromosomes align along the equator of the cell. So, and the fourth in the meiosis stage was anaphase 1. So, anaphase 1 here, each of the homologous chromosomes get pulled towards opposite poles of the cell as the spindle fibers retract. This equally divides the DNA between the two cells which will be formed. And for the 5 and 6 stages of meiosis, which is telophase 1 and cytokinesis 1, during telophase 1, the nuclear envelope reforms and spindle fibers disappear in cytokinesis 1. The cytoplasm and cell divides, resulting in two cells that are technically haploid. There is one chromosome and two chromatids for each chromosome. Here, each of the homologous chromosomes get pulled towards opposite poles of the cell as the spindle fibers retract. This equally divides the DNA between the two cells which will be formed. Meiosis 2. Prophase 2 and prometaphase 2. These stages are identical to their counterparts in meiosis 1. Metaphase 2. In metaphase 2, chromosomes line up in single file along the equator of the cell. This is in contrast to metaphase 1, where chromosomes line up in homologous pairs. Anaphase 2. Next, sister chromatids are pulled to opposite poles of the equator. Telophase 2. This stage is the same as telophase 1. Cytokinesis 2. Again, in the cytoplasm and cell divides, producing two non-identical haploids daughter cells. As this is happening in both cells produced by meiosis 1, the net product is 4 non-identical haploid daughter cells, each containing one chromosome consisting of one chromatid. These are fully formed gametes. And that's it for my instructional video. I hope you learned. Thank you.